And so I'm recording. Yesterday, we left off just building a glossary about the muscular system and what that's going to entail. Today, I'm going to go over how a muscle cell contracts, how that whole process works. And this is a long story, but it's an important story that you're going to need. You're going to need to be able to understand. I'm not putting essay questions, open-ended essay questions on these quizzes, as you know, but in my opinion, it would be healthy for you to determine whether or not you understand this topic if you can explain this to someone else. If you don't have somebody else to explain it to, or if no one else will want to listen to it, you can try your best to write out the steps of this process. So using the glossary and all the terms that we developed yesterday and wrote down and definitions and all this business, now this next section in your book is, I believe it's called the sliding filament model of contraction. So those in a really brief 10-second explanation, those thick and thin filaments that we described yesterday and defined, what's going to happen is they're going to slide past each other like my fingers, like this. And that's how the muscle cell actually shortens. I'm going to give you the really nitty-gritty and all the details of how that happens. Your book does too. Hopefully, if I walk you through it step by step, it's just a story. This happens, then that happens, then that happens. It's just a series of events. But if you listen to me explain it, hopefully you'll understand it better. So that's where I'm headed to right now. So we're going to use all of the business that we defined yesterday to just tell this story. So what we're doing is we're describing completely the events at the neuromuscular junction and what we call the excitation contraction coupling. So this is the sliding filament model of contraction. So let me get a pen. What I'm going to draw now is a nerve ending. The ending of a neuron. A neuron is just a nerve cell that transmits an electrical impulse. Under heavy magnification looks something like this. A muscle cell, as it relates to this nerve ending, looks something like this. A muscle cell, as you know, is long and cylindrical. I'm going to make it extra wide and fat because we have to fit a lot of stuff in this picture. So this is the skeletal muscle cell. I'm just going to write muscle fiber. This is the neuron. Technically, it's called the axon terminal of the neuron, but we'll learn that in the nervous system next. So here's what happens. I'm going to say some things and I'm going to draw a picture as I say them. First, an action potential travels down the axon membrane, this neural membrane. So action potential, you don't need to, that's just, it should be interpreted in your mind right now as an electrical impulse. So action potential, electrical impulse travels down this membrane. That's what I'm drawing now. When it reaches the end of the neuron, it causes extracellular calcium out here to influx into the nerve cell. Okay? So this electrical impulse causes calcium ions to influx into the neuron. Calcium ions then are responsible. These bubbles that I'm drawing right now are just storage vesicles for chemical called neurotransmitter. So the dots inside of those bubbles are a chemical. Generic name for that nerve chemical is neurotransmitter. In this case specifically, all skeletal muscle fibers use a specific neurotransmitter called acetylcholine. Sorry, I'm having trouble writing on this board this morning. That's supposed to be a C, A-C-E-T-Y-L, C-H. It's a long word. That's just a chemical that is used to transmit an impulse from the nerve cell to the muscle cell. When calcium flows into this nerve cell, it causes this storage vesicle of acetylcholine to migrate to the end and release its contents by exocytosis. So electrical impulse, action potential, calcium influx, release of acetylcholine into this space, which is called a synaptic cleft. The word synapse just refers to this whole junction between nerve and muscle cell. So the space here is called the synaptic cleft. There is no physical contact between the nerve cell and the muscle cell. There's this space. Acetylcholine diffuses across the synaptic cleft and binds to receptors on the motor end plate of the muscle fiber. This indented portion of the muscle fiber is called the motor end plate. I'll go through this whole story again when we get to the end. So acetylcholine diffuses across, it binds to these receptors that I've drawn, these rectangles. When the chemical acetylcholine binds to these receptors on the motor end plate, it initiates an electrical impulse or action potential along the sarcolemma of the muscle fiber. So it goes electrical, chemical, electrical again. So now the muscle sarcolemma, the muscle plasma membrane, is exhibiting an electrical stimulus or action potential of its own. That electrical stimulation or impulse needs to be guided toward the interior of the cell, and that's done by T tubules, which is in your glossary of terms that we went over yesterday. So these tubes that I'm drawing that go to the inside of the cell are T tubules, and they get their name because the top of them do this. They kind of look like T's, like this. So the electrical impulse is guided inward by these T tubules to the inside of the cell, as so. Associated at the end of a T tubule, associated with each T tubule, are sacs of the sarcoplasmic reticulum. Okay. So these yellow things that I'm drawing, they have a fancy name, but they're just storage sacs for calcium ion. And they're connected to each other like so. So that's, I'll write out what I'm drawing in yellow here. These yellow storage containers are called terminal cisternae. We went over this yesterday at the end. They're called terminal cisternae of the sarcoplasmic reticulum. So everything in yellow technically is the sarcoplasmic reticulum. The bulging sacs that I've drawn at the end of the T tubules are called terminal cisternae of the sarcoplasmic reticulum. So let me just label that. Singular would be cisterna. I'm just going to write of the SR. Just think of those as storage containers for calcium ions. Now, the term triad, which you will read in your textbook, just refers to what's going on at the end of a T tubule. Notice that I've drawn 2 terminal cisternae associated with each T tubule. 2 plus 1 is 3, so we call this area the triad. I'm going to go over this whole story again when I get to the end. So when this electrical impulse travels down the T tubule and gets to the triad area, it causes calcium ions to be released from the terminal cisternae of the sarcoplasmic reticulum. And calcium ions then flood the interior of the cell. Once calcium is released from terminal cisternae, I'm going to say this. This is the first. There's two parts to this story. This is the first part, first chapter. The second chapter I'm going to do on my next screen. So I'm going to go over this first chapter really quickly right now. Ready? First, an electrical impulse called an action potential travels down the axon. When it reaches the end of the axon of the neuron, it causes calcium ions to flow into the nerve cell. Those calcium ions are responsible for dragging this vesicle that has stored acetylcholine in it to the end of the nerve cell. That vesicle releases acetylcholine. The acetylcholine chemical diffuses across this space called the synaptic cleft and binds to receptors on the motor end plate of the muscle fiber right here. When acetylcholine binds to receptors on the motor end plate of the muscle fiber, it initiates an action potential, electrical stimulus, along the sarcolemma of the muscle fiber, like this. That action potential, electrical stimulus, is guided inward via these T-tubules. When that electrical impulse reaches the triad area, calcium ions are released from the terminal cisternae of the sarcoplasmic reticulum, and calcium floods the interior of the muscle fiber cell. That's chapter 1. Chapter 2 is we're gonna magnify this. So even under heavier magnification, we're gonna look at some smaller things that are going on. Now I'm gonna draw a myosin molecule that we drew yesterday, a thick filament myosin. There's the M line. I'm not going to gum up my picture with an entire sarcomere that we went over yesterday. Just going to simplify this and draw this one red. I'm using red because that's what your book uses. This is a thick filament. This is what's going on deep inside the muscle cell, okay? Actin, I think your book uses blue, so I'm going to try, different books use different color. This is the thin filament, actin. There is active sites on actin that the myosin head can attach to. They're not all on the exact same side. They kind of roll around. Those are active sites that this myosin head here can reach up and grab the actin molecule. However, there is another polypeptide called tropomyosin that covers these active sites, and it wraps around actin like a grapevine. It's just like a branch, and it covers the active sites, so myosin can't reach up and grab actin, bind to it. On tropomyosin are these little tabs. They're like switches. Those little yellow tabs are called troponin. These were all part of our making up a dictionary or a glossary yesterday. All right, so here's what happens. I'm going to just remind you really quickly. We went through chapter 1. We started with a nerve impulse up here. We ended up with releasing calcium ions inside of the muscle cell. Here's what that calcium does. When calcium is released, to the interior of the cell from those terminal cisternae of the sarcoplasmic reticulum, calcium ions bind to that troponin. When calcium ions binds to troponin, it lifts tropomyosin off of actin, exposing these binding sites. So I'll say that again. Calcium binds to troponin, lifting tropomyosin off of actin, exposing actin's binding sites. Now myosin can reach up, grab the actin molecule, and if this myosin head is energized by an ATP molecule, the myosin head can pull actin toward the M line this way. That's how a muscle cell, an entire muscle cell, when energized or innervated by a nerve cell, when it contracts, it gets shorter like this. And it's because all of these actin molecules in every sarcomere are being pulled toward the M line like this. And then in the next sarcomere, the actin molecules will be pulled toward their M line like this. And that happens thousands and thousands of times within this muscle cell. It just, it pinches that cell toward all of the M lines of these sarcomeres, all the way down the muscle cell. That's what causes it to short. So I'm going to go through this one more time with you, and then we're going to move on. So I'm going to go back to my original picture. Ready? Chapter 1. It's important. I'm going to expect you to know the sequence of events. And your book explains it, but now you know the whole story in a bit simplified fashion. So first, nerve impulse action potential travels down the axon. When it reaches the terminal end, it causes calcium ions to flow into the neuron. That causes a neurotransmitter, acetylcholine, that is stored in these vesicles to be released into the synaptic cleft. That acetylcholine diffuses across this space, the synaptic cleft, and it binds to receptors on the motor end plate of the muscle fiber. When acetylcholine binds to receptors on the motor end plate, it initiates an action potential along the sarcolemma of the muscle fiber. That action potential is guided inward via a T-tubule. When it reaches the triad, the end of the T-tubule, calcium ions are released from storage from the terminal cisternae of the sarcoplasmic reticulum. Then calcium floods the interior of the cell. Calcium ions then bind to troponin, which lifts tropomyosin off of actin, exposing the binding sites, the active binding sites on actin, so that myosin can reach up, grab the actin molecule, bind to it, and pull it toward the M line. And that also, the same thing is happening over here. This myosin molecule would reach up at the same time and pull its actin molecule this way. So the muscle cell shortens during contraction, and that develops tension or some sort of force of energy that the muscle can pull on a bone, for example, and move it. Before I leave this story for good today, and I can repeat this tomorrow if you'd like, no problem, but before I leave this for good, there's one last thing I'm going to say about this story. Back here in chapter 1, after, whoops, I was going to use a black marker. After acetylcholine is released from these storage vesicles and diffuses into the synaptic cleft. Right here, there is an enzyme that lives in this area of the synaptic cleft. That enzyme has got a long name, but it begins with acetylcholine. So I'm going to write that enzyme name out. It's called acetylcholine esterase. That long, super long name is just an enzyme that degrades acetylcholine, okay? Whenever anything ends in ASE, you can bet your life that it's an enzyme. So that's a dead giveaway. And the name acetylcholine tells us what it does. It breaks down this chemical. That enzyme lives in this space, this synaptic cleft. And I'm going to draw a little Pac-Man head. What it does is it gobbles up this enzyme. It degrades it. It breaks it down. So as soon as this neurotransmitter, acetylcholine, is released into this space, some of it diffuses across this space and binds to the muscle cell, the motor end plate receptors, initiating all that business of contraction. But we don't want a muscle to be continually in a contracted state. One of the things that allows this muscle fiber then to relax is this enzyme quickly gobbles up any excess neurotransmitter in this space and degrades it so that this muscle doesn't stay in a continued state of contraction. So I did want to mention, and I want you to recognize, this enzyme is partly responsible for relaxation of this muscle by degrading this chemical, acetylcholine, in the synaptic cleft. All right, I know that that was a lot. Please read your book through those steps multiple times. I'm happy to answer any questions you'd like. Now we need to go on to something called the cross-bridge cycle. And as always, I record these meetings and I post them. Usually by the end of the same day, I post them to the module on the Canvas page. So if you need to review this, go ahead and do it. This cross-bridge cycle, that's how it's titled in your book. Just think of this. The myosin head and ATP. Remember, I just told you at the end of that part of that story, the myosin molecule, this thing, reaches up and grabs actin and pulls it toward the M line. How this myosin molecule gets energy to do the pulling is from ATP. That's how it gets its energy. I'm going to describe to you, and your book describes this to you, but it might be confusing if you just read it without explanation. So I'm going to go through step by step how ATP gives myosin head its energy. And your book refers to that as the cross-bridge cycle. Okay? So I'm going to draw a picture, and we're going to go through this step by step. Here's a myosin molecule. Here's an ATP molecule inside the muscle cell. Step 1, ATP attaches to the myosin head. Step 2, ATP splits its terminal phosphate. Remember, ATP has 3 phosphate, triphosphate. It has 3 of these phosphate molecules attached to it. When we cleave or split that phosphate off of the ATP molecule, it transfers energy from stored energy from ATP to another molecule. In this case, it's going to transfer energy to the myosin molecule. So when ATP splits, it looks like this. The myosin head gets energy, so it moves its head this way. And attached now to the myosin head, whoops, I wanted black, is ADP, adenosine diphosphate, plus P. So we've just broken one of the phosphates off of ATP, and we're left with ADP and P, but they're still attached to the myosin head. That causes the myosin head to go from this position this way. It's like pulling back that little thing on a gun, a handgun, I don't even know what it's called, that sort of cocks a gun. So we're cocking the head of the myosin molecule. Now, myosin, after it gets energy from the splitting of ATP, its head looks like this. ADP and P are still attached to it, but now it reaches up and it grabs the actin molecule because calcium has exposed the active binding sites, and ATP, the splitting of ATP, gave it the energy to pull the actin molecule in this direction. So it attaches and it moves, the myosin head moves from this back to this position after it pulls. Sorry, I wanted blue. So here it's just pulled the actin molecule a bit this way. Guess what? The ADP plus P are still attached. So after this pulling, I am going to write this out because I'll probably use this language on the next quiz. The pulling action is called the power stroke of muscle contraction. So that term power stroke just refers to this action of pulling actin toward the M line. That's what this term means. So after that, a new ATP, during the power stroke, ADP and P are released, but the myosin molecule is still attached to the actin molecule. It can't release. The myosin molecule cannot release from actin until a new ATP attaches to it. So when a new ATP attaches here, after these come off, a new one attaches, then myosin releases from actin. It has a new ATP attached to it, and it's released from actin like this, and the whole process can start again. So I'm going to go through this verbally with you really quickly again. First, ATP attaches to a myosin head. Then ATP splits its terminal phosphate and cocks the myosin head, giving it energy. And ADP and P are still attached to the head. The myosin head then reaches up, attaches to actin binding site, and the power stroke of pulling actin toward the M line occurs. Then ADP and P are released. A new ATP molecule attaches to myosin. Myosin head can now release from actin, and the whole process can start over. If we need a sustained contraction, for example, if you're working out and you lift up a weight and you want to hold that contraction for a long time, this cycle just keeps recycling. As long as we have enough ATP, this can just keep going and keep going and keep going until the muscle fatigues and it can't sustain that contraction any longer. I don't know if you've ever heard of the term, I'm sure some of you have, rigor mortis, which is the continued state of skeletal muscle contraction after we die, after a person dies. This whole process sort of explains how rigor mortis occurs in people. So what happens in a person that just dies or is deceased recently is what ADP is in that person's, stored ADP is in that person's muscular system. It's used to contract the muscle. And because the person is no longer breathing, new ATP molecules cannot be generated in that person that just died. And so this can never be released. The myosin head, because a new ATP molecule is not available, because the person is no longer breathing, this binding of myosin to actin cannot be released. It doesn't let go. And so rigor mortis is the continued state of muscular contraction. And that usually lasts for anywhere from 20 to 30 minutes until the person's entire muscle tissues start to break down. Once that occurs, once the myofilaments start to break down and proteins start to break down, then all of that process start to occur. The person, the recently deceased person's muscular state is in a continued state of contraction because no new ATP molecules are available. All right. So now that you understand at least a little bit how a muscle cell can contract, a skeletal muscle cell, I'm going to write the duration of contraction of any muscular skeletal muscle contraction depends on three things. And I'm just going to list them as 1, 2, and 3. So what dictates how long a muscle can contract is first dependent on the duration of stimulus from the motor neuron, from the nerve cell. So way back in our first drawing, way back here, number one, how long this nerve cell keeps stimulating this muscle cell dictates first how long we can keep contraction. Sorry, I didn't go back to this board I wanted to go back to. Second, the presence of free calcium ions in the sarcoplasm of the muscle cell. So we need calcium. First, we need neural stimulation. Then we need the presence of free calcium in order for those active sites on actin to be exposed. And finally, we need availability of ATP molecules. Those three things dictate how long a muscle can stay contracted. If this shuts off, the muscle cannot stay contracted. If any one of these gets depleted or whatever, then the muscle cannot stay contracted. All right, as we go on, I need to define two different types of contraction, isotonic versus isometric contraction. Trying to get through as much of this as I can. I know I can't hit everything in your book, but I try to do as many major points as I can and simplify them for you so that when you do read your book, I'm not naive enough to believe that every single student's going to sit down and read their textbook prior to these meetings. I get that. That's not realistic. It doesn't matter to me when you read your book. It's if you read your book. I'm going to say this because it's true. From my experience of teaching A&P for a very, very long time, students who read their textbook always do better than students who do not read their textbook. Whether or not you think you're absorbing it, you are. So let's go on to these two different. This one, isotonic, I'll define it and then give you a couple different types. What this means in definition is just the muscle changes in length during contraction, okay? And during contraction is when it develops force or tension, both tension and then producing force. That just means the ability to do some work. So isotonic, key is muscle changes in length. There's two different types of isotonic contractions. I'm just going to list them. I don't have room to write the definitions out, but I'm going to list them and I will state their definitions for you. A concentric isotonic contraction is when the muscle length shortens. That's typical. So let's just go to the analogy or the example that you're working out with weights. If you do like a bicep curl with a weight, that flexion at the elbow, that would be a concentric isotonic contraction. The second type is eccentric. I said I didn't have room to write this, but I do. It's when the muscle lengthens. The muscle, it lengthens, gets longer, doesn't get shorter. So it's the opposite. So when you're doing a bicep curl, as your arm bends, I'm just going to do this. Ready? If I'm doing a bicep curl with my arm, as I do this, my biceps muscle shortens. Okay, that's a concentric isotonic contraction. If I still have the weight in my hand and I do this with the weight, my muscle is lengthening, but my arm is still moving. It's still changing. That would be an eccentric isotonic contraction. All right. So now we were doing isotonic versus isometric. An isometric contraction is when the muscle does not change in length, but force is developed. Both force and tension are developed in the muscle anyway. We've all experienced this. If you try to lift something that you're unable to lift, if you go out and try to lift your car, for example, off the ground, you can't do that, obviously. But force is still developed in all of the muscles as you're trying to lift. So if you try to move the wall of your house by pushing against it, it's not going to move. And you develop force in your muscles, but you're not you're not moving the load, but force is still developed. And the load is not moved. In an isotonic contraction, we actually move the load that we are trying to move, like lifting the weight. So tension is created during an isometric contraction, but the load is never moved because it weighs more, or we are unable to move it based on the force that we can develop with our muscles. So I'm going to switch topics here. I'm trying to hit as many as I can. Remember at the very beginning of this business with skeletal muscle tissue, one of its characteristics is that it is multinucleate. This is going to sort of explain how an adult or mature skeletal muscle cell can become multinucleate. And we're going to first start off with this term, myoblast. We know what the term blast means. It means immature cell. We know what myo means. It means muscle. So by definition, this is a new, young, immature skeletal muscle cell. All right? And they give rise to mature muscle fiber cells They give rise to mature muscle fibers. And here's what they do. Myoblasts fuse together in a young state. to form a mature fiber. That's how they become multinucleate, because myoblasts each have their own nucleus. And when they fuse together, the nuclei don't fuse together. They stay separate, and we end up with a multinucleate mature muscle fiber. Now, not all myoblasts fuse together to form a mature fiber. Some, I'm just going to write this out, myoblasts that do not fuse at a young early stage become myosatellite cells. That's supposed to be SATELLITE, myosatellite cells. And these cells specifically aid in muscle repair after damage. All right, the next topic in your book is quite complicated. I know I'm going through a lot of complicated topics today, but let's see, today's Thursday, so we're going to lecture today, tomorrow, and then have another quiz again, I believe, on Monday. So I want to cover as many things as possible. Before I leave today, I'm just going to cruise through this. Muscle, that's supposed to be a T, muscle metabolism. When you read this term in your book, just think this: how a muscle acquires ATP. Really quickly, Bio 1 is a prerequisite for this course. Here's one big reason why. Because it's imperative that if you haven't had Bio 1 for a long time, don't freak out. I will give you the nitty-gritty of it once again, but if you understand that awful thing that you had to learn in Bio 1 called cellular respiration, both glycolysis and cellular respiration, it's really going to help you during this time, okay? So first and foremost, muscles, they do store, but very little. They store very little ATP. Some, but not a lot. They store about 4 to 6 seconds worth of contraction. So we're going to start with this story like this. Our muscle cells, our skeletal muscle cells, do store ATP, but only enough ATP to provide for about 4 to 6 seconds worth of contraction. Obviously, if we want to continue a muscle contraction for longer than 4 to 6 seconds, our muscle cells have to get ATP somewhere else, some other way. And ATP can be generated in 3 ways. That's what muscle metabolism is about. First, the first way, and by the way, a muscle cell uses these steps in this order. It's called direct phosphorylation. So before I go any further, when a muscle cell contracts, the first thing that it does is it uses stored ATP. Always. Step 1, when a muscle begins to contract, it uses what little ATP it has in it stored. Second thing it will do are these 3 steps. And the first one of those three steps is direct phosphorylation. It is exactly what it says. It is anaerobic, so we don't need oxygen, anaerobic, A-N, to do this. When we use up ATP, here's an ATP molecule. When we break that terminal phosphate, what's left? ADP plus P. So when a muscle uses this stored ATP, this is what it's left with. Direct phosphorylation just means we're going to directly stick a P back onto this ADP to make ATP. So I'm going to write that out. Adenosine diphosphate because this ATP has been used, adenosine diphosphate, in order to make a new ATP out of this, we're going to have to add a phosphate back to it. It's not as easy as just pasting it back on from where it came from. We have to use another source. And that other source is called creatine phosphate. You might have heard, if you're a workout person, you might have heard that creatine energizes muscle building. This is where that comes from, that theory comes from. This is just a molecule that has a phosphate on it. We're going to steal this phosphate and stick it onto this ADP. So we draw that chemical reaction like this. We've just taken this phosphate on creatine phosphate, stuck it back onto ADP to get ATP, and we end up with creatine without its phosphate, because we've used this phosphate, stuck it on here, and we get now a new ATP. The body can't do that very quickly. There's an enzyme involved that speeds up this reaction that's really important, and it's called creatine kinase. So creatine kinase enables the process of direct phosphorylation to occur in a muscle cell. So that's step one. The second thing, let's say, by the way, I'm going to tell you, this provides, direct phosphorylation provides about 15 to 20 seconds worth of contraction. So now we're all the way up to a whopping 20 seconds. If we want to sustain a contraction, first our muscle does this, then it does this. Let's say we want to continue the contraction. Our body needs to continue a muscular contraction. The second thing a muscle cell does to regenerate or make new ATP is called even though direct phosphorylation was anaerobic, this is called the anaerobic pathway. You learned it in Bio 1 as glycolysis. I don't have time to go through the whole glycolytic pathway. I will give you the key points and remind you that this is, anaerobic says no oxygen necessary, no oxygen needed. When we have a cell, any cell in the human body, this is a quick review of glycolysis. Glucose is a simple sugar that we can use for an energy source really fast. That's why it's always used for these examples. The word glycolysis just means glucose, lysis, to break down a glucose molecule. That occurs, let's just say this glucose molecule is in our bloodstream because we just ate a candy bar, whatever, it's available for us to use, our cells to use. When this glucose molecule enters a cell, the process of its breakdown occurs at that point. Glycolysis occurs here. And it does not require any oxygen to occur. And what it does is it initiates the beginning of breaking apart this glucose molecule, and it gives off as a byproduct 2 ATP molecules and lactic acid. I'm going to put in parentheses. Actually, it's pyruvate that gets converted to lactic acid if there's no oxygen available. Okay, so we've just made 2 ATP. If this were a muscle cell, guess what? We quickly converted glucose into 2 ATP molecules. We can use this now to sustain or continue our contraction. So we've got glucose breakdown from either a blood source or glycogen stores in the body, but we've got glucose breakdown. When it enters the muscle cell, In the absence of oxygen, that process of glycolysis occurs, and we harvest 2 ATP and pyruvate, which gets converted to lactic acid. All right. This anaerobic pathway produces very little ATP, but it produces it very fast. So what we say is it's not really efficient. So it's fast, but its drawback is only 2 ATP molecules per glucose molecule. And this can provide us for about 30 to 40 seconds worth of contraction. So now we're up to a whopping 40 seconds. We still haven't used any oxygen though. So your body does all of these things without you even having to breathe, really. You know that if you've ever tried to sprint anywhere, across your backyard, down the street, whatever, you chase your dog for 50 feet, you don't have to really breathe to get that. It's an explosive type of energy that produces not a very long contraction. You can't sustain that without breathing for very long, but you can up to about 30 or 40 seconds. So let's just take that famous sprinter Usain Bolt as an example. When Usain Bolt starts out of the blocks when he's racing and that gun goes off, the first thing his muscles do is they use stored ATP for contraction. That gets him about 4 to 6 seconds down the track. Then his muscle cells do this. They direct phosphorylate to produce ATP molecules. That gets him about 15 to 20 seconds, and he's so fast that he's done running by now. But let's say he wants to go a little further. Then his cells do this, the anaerobic glycolytic or glycolysis pathway. That will enable him to run about 30, 40 seconds, sprint for 30 to 40 seconds without really having to breathe. He can exhale the whole way if he wants. And the anaerobic process is, in the absence of oxygen, breaking down a glucose molecule to get 2 ATP. And these are the byproducts. Now let's say Usain Bolt wants to continue running for a half hour. Well, the body has to do what's called the aerobic pathway or cellular respiration to do that. So I'm going to draw a similar picture on my next page, and this is the last thing I'm talking about. This is how ATP is generated the third way. This is the aerobic pathway that you learned in Bio 1. It's called cellular respiration. It gets the name respiration because it uses oxygen. So O2 is required. And this is what our muscle cells do. I'm going to draw a round cell just because I can fit more things into it, even though we know skeletal muscle cells are long and skinny. So I'm going to quickly draw this. We're going to use glucose again as an example. When it goes into the cell, we get glycolysis, anaerobic, 2 ATP, no oxygen during this stage. We also got pyruvate. And if there is no oxygen present, that pyruvate gets converted to lactic acid. If there's no oxygen. If there is oxygen, in other words, if we start breathing heavily, oxygen comes into the bloodstream via our respiratory system. That oxygen can also diffuse into the cell and into a mitochondria in that cell. And skeletal muscle cells are chock full of mitochondria. So oxygen then heads to the mitochondria. This pyruvate also heads to the mitochondria. That's where cellular respiration occurs, the electron transport chain, the Krebs cycle, and the electron transport chain. I'm not going to go through those steps, and I'm not going to ask you about those steps on the quiz, by the way. But I do want you to know that if we need to sustain a longer contraction, we have to breathe. That oxygen comes into the cell, heads to the mitochondria. This byproduct of glycolysis, if there's oxygen in the cell, also heads to the mitochondria. Those two components get broken down and give off a whopping 32 ATP. Actually, that includes this out here. But 32 ATP molecules per glucose can be made. So this is slow. This takes a long time to occur relative to the other things happen in milliseconds, the other reactions. This is slow, but we say it's very efficient because it produces 32 ATP per glucose molecule. So that's how our body can keep chugging along. If we want to go for a 3-mile run or jog, you know that you can, if you're a jogger or a runner, even if you're not, you know that if you run for very long, after about 30 or 40 seconds, you can try to hold your breath. You have to start breathing. Your respiratory centers in your brainstem force you to start breathing because of the buildup of CO2. And you start taking in oxygen heavily into your body. That's to sustain this activity. In order to sustain muscle activities, we need a lot of ATP. And the only way our cells can do that is by breathing oxygen, and we also need some sort of an energy source. That's a whole different conversation of what energy sources. We usually use these glucose molecules first, and then our body will, if there's no starches or glucose to be broken down, then our body goes after protein and fat, which is why, by the way, it's so hard to lose weight, stored fat, because that's usually the last place our body goes in order to conduct this process. The last thing I'm going to say a few things about this. We can sustain this really, literally for hours until our muscles fatigue. As long as there's an oxygen source, and as long as we have an energy source of some sort, we can sustain. We can't sustain it forever, obviously. I'll talk about muscle fatigue later if I have time. It's not really determined scientifically. There's different things that cause different types of fatigue. Anyway, you know that we can't sustain muscle contractions forever, but we can for hours as long as there's this source and this source to produce, to keep cranking out these ATP. Now, let's just backtrack for a second. I'm almost finished. If we don't have oxygen, this does not occur. The aerobic pathway, it can't occur if we don't have oxygen. So let's go back really quickly. I wanted to say a byproduct to glycolysis, this one, if there's no O2, our body makes pyruvate, which will not get converted to ATP because it has no oxygen to combine with. So we make this. We make lactic acid builds up in our muscles if there's no oxygen available. So picture this. Let's just say I told everybody to stand up right now, bend down, touch the floor, and then jump up and try to touch the ceiling. And do that as long as you can without breathing. You could sustain that for roughly 20 to 30 seconds. And you know that you'd have to start breathing in order to sustain anything. This builds up during that 20 to 30 seconds, lactic acid. That's what gives us the burn. The buildup of lactic acid in our muscle cells gives us that burning sensation inside of the muscle cell while we are performing that anaerobic exercise. As soon as we stop that exercise and start to breathe, this lactic acid gets broken down very quickly. A lot of it gets transported to the liver. And the liver breaks down that lactic acid, and we no longer feel that burn inside of the muscle cell. So if you are active physically, and I highly recommend you should be, in any way, and you do, even if you go up 3 flights of stairs, your leg muscles start to burn. And that's this building up in your muscle cells. As soon as you get to the top of the stairs, it only takes about 5 to 10 seconds for that burn to go away. That's this being transported to the liver and the lactic acid being broken down very quickly by the liver. All right, I've gotten through a lot of business today, and I appreciate your attention. So I'm going to cut off my recording, and then you can ask me anything you'd like.